Bismillahirrahmanirrahim. Our today's topic is transport of carbon dioxide in the blood and tissues. So this is another very important topic in relation to your exams and your particularly the practical field when you apply physiology in your daily routine. So in previous lecture we discussed the transport of oxygen in the blood and tissues, while today we will see and discuss carbon dioxide. So why carbon dioxide is important? Because even in most abnormal conditions means that when oxygen and carbon dioxide perfusion ratio is disturbed. So in that condition, even then, however, the amount of carbon dioxide in the blood has a lot to do with the acid-base balance of the body fluid. So obviously we have discussed it in many instances, the buffer system of the body, of the blood. As you know, that blood has a pH of 7.4 plus minus 0.5. So it means that the range is 7.35 to 7.45. So this acid-base balance, if this pH goes above 7.45, it means that alkalosis is there. While if this pH comes below 7.35 and as in many diseases like metabolic or certain respiratory diseases, so this pH may drop down to 7.25 or 7.3 or 7.2. So this is called acidosis. So because of carbon dioxide, obviously if you just inhale oxygen, take a deep breath just now while listening to this lecture, simple, the amount of carbon dioxide comes out of your respiratory system will be based on the amount of oxygen that is coming in, or the amount of oxygen that goes inside your body will depend on the amount of carbon dioxide that comes out. This is called ventilation perfusion ratio. Just think that there is some disease, there is some condition in which carbon dioxide, actually the basic stimulus for breathing is carbon dioxide. It is not oxygen. It is never oxygen. The stimulus for breathing or ventilation or respiration is carbon dioxide, because carbon dioxide is a waste product. So waste product, body needs to remove it. So as carbon dioxide, as you are respiring, as your cells are respiring, they are producing carbon dioxide. That carbon dioxide is coming into the blood, and the blood wants to get rid of that carbon dioxide. So this actually makes sense because that carbon dioxide which is piling up in your blood needs to be removed out of your blood. So how it is removed, that is our topic of today's discussion. So, but meanwhile, that carbon dioxide that is present in blood will actually combine with the water in plasma or plasma proteins, and it will form carbonic acid. So just think for one moment, greater the amount of carbon dioxide, if there is, again coming back to that particular point, greater the quantity of carbon dioxide that is present in your blood will react more with water and will make more carbonic acid. So carbonic acid will accumulate in your blood, and where goes the pH? pH will drop down from the normal 7.35. So this will increase and the pH will decrease from 7.35. So that is the scenario. The scenario is this. So simple. The quantity of carbon dioxide is like the amount of carbon dioxide in the blood has a lot to do with the acid-base balance, and the carbon dioxide can usually be transported in far greater quantities than oxygen because body does not want to accumulate it. And under normal resting conditions, like you're listening to this lecture or you are just having a cup of tea in a resting mode, an average of 4 ml of carbon dioxide is transported from the tissues to the lungs in each 100 ml of blood. So there are certain types, there are certain physical forms in which carbon dioxide is being transported from tissues to the blood and from blood to the outside of the body. There are 3 states in which carbon dioxide is transported out of body. One is within the blood. The first, as dissolved carbon dioxide in the plasma, it is 21 times more soluble in water as compared to oxygen. So that is one way by which carbon dioxide is being transported. Second is carbonyl hemoglobin. So hemoglobin not only transports carbon dioxide, but it also not only transports oxygen, but it also transports carbon dioxide out of the body. So just remember when you are asked the function of RBCs, so RBCs also transport carbon dioxide from tissues to blood, blood to lungs and into environment. And the third and the most important way by which, or this you can say the primary transport mechanism of carbon dioxide is bicarbonate ion, that is released when carbonic acid dissociates. So these are the 3 forms. Number 1, dissolved carbon dioxide. Dissolve is simply dissolution. You take carbon dioxide and add it in water. Then second is carbonyl hemoglobin, and if you see the diagram of all these 3 ways by which carbon dioxide is, this is a carbon dioxide which has to be disposed of, which is being made in the body or in tissues in response to cellular respiration. So cells are respiring, they are producing carbon dioxide. So carbon dioxide can be dissolved, CO2 in physical solution can be dissolved in water of blood and simply goes out, comes here, and these are alveoli, comes here and simply diffuses out. That is number 1. Number 2, here carbon dioxide combines with, here what happens with water. Carbonic anhydrase is there. Carbonic anhydrase actually causes dissociation into acid and base. This bicarbonate ion gets here and it is again transported and shifted to carbon dioxide and goes back simply. Then the third one is here carbonyl hemoglobin, NH2. And it is replaced by COO, that is COO, and simply this is taken to the alveoli. And so the first is carbon dioxide in dissolved state. PCO2 or partial pressure of carbon dioxide of venous blood is 45 millimeter of mercury, and that of arterial blood is 40 millimeter of mercury. It means that the blood that is contained in the vein or in artery has different amount of partial pressure of carbon dioxide, respectively 45 millimeter of mercury and 40 millimeter of mercury. So only about only about 0.3 ml of carbon dioxide is transported in the dissolved form by each 100 ml of blood flow. So this is about 10% of all the carbon dioxide normally transported. So second one is carbonyl hemoglobin. So we have already seen that how it is being transported. This is again a reversible reaction because loading and unloading has to be taken place, and there is a loose bond, so that carbon dioxide is easily released into alveoli where PCO2 is lower than in pulmonary capillaries. So a small amount of carbon dioxide also reacts with same way with plasma protein in the tissue capillaries, that is a negligible transport. While CO2 as carbonic acid. So this is a primary mechanism. The dissolved carbon dioxide in the blood reacts with water to form carbonic acid. This reaction would occur much too slowly to be important were it not for the fact that inside the red blood cell is a protein enzyme called carbonic anhydrase. Carbonic anhydrase is a very important enzyme. It has many therapeutic implications as well. You will see in second prof. So carbonic anhydrase is present, and it actually catalyzes this reaction, carbon dioxide plus water, and this carbonic anhydrase accelerates this reaction rate about 5,000-fold. So instead of taking minutes or seconds to complete this reaction, gets equilibrium within a very small fraction of a second. So dissociation of carbon dioxide, so in one fraction this reaction takes place, carbon dioxide plus water making carbonic acid. So in another fraction of second, the carbonic acid formed in the red cells dissociates into hydrogen and bicarbonate. So this again is here like this. This is carbonic acid and this is dissociated into hydrogen ion and bicarbonate. So that is why you can see increase in carbon dioxide quantity in the blood will cause acidosis, will decrease the pH towards the acidic side. So most of hydrogen ions then combine with the hemoglobin in the red blood cell because the hemoglobin protein is a powerful acid-base buffer. So in turn, many of the bicarbonate ions diffuse from red cells into plasma, while chloride ions diffuse into red cells to take their place. So that is here, you can see this. So what actually happens? Bicarbonate ion diffuse from red cells into plasma, while chloride ions will diffuse into RBCs in place of bicarbonate. So what is the situation? Situation is that first of all, what we are currently discussing, we are discussing transport of carbon dioxide in RBCs. So once carbon dioxide is produced, carbon dioxide will come into RBC. Available sites for hemoglobin will be taken up by carbon dioxide, and excess of carbon dioxide which cannot be attached to hemoglobin will actually react with water that is present in. So ultimately this will make carbonic acid in the presence of carbonic anhydrase. So once a carbonic acid is produced, simply it will dissociate into bicarbonate and hydrogen. So hydrogen ion will not be released into the blood because it will disrupt the pH until and unless the quantity is immensely overwhelming, or there is a greater quantity of hydrogen ion which this buffer, one buffer is this and one buffer is present within the RBCs. So RBC's buffer system or pH neutralizing system will actually take up those hydrogen ions. but bicarbonate ion is free. So bicarbonate ion will move out into the blood, and to take that place, this Cl will go inside, because hydrogen ion is there, so Cl will go inside. So simply this bicarbonate will come out. So now this chloride is being moved out. So thus the chloride content of venous RBCs is greater than that of arterial red cells, a phenomenon called the chloride shift. Just remember that. So this method accounts for about 70% of carbon dioxide transported from the tissues to the lungs. So thus this means of transporting carbon dioxide by far most important. What about rest? The quantity of carbon dioxide that can be carried from the peripheral tissue to the lungs by carbamino combination with hemoglobin, and carbamino means carbon dioxide and amino group or NH2 group. So plasma proteins is about 20% of total quantity and 10% in the dissolved state. So now we have the breakup. In dissolved state it is transported about 10%, in carbamino hemoglobin around 20%, while with bicarbonate ion it is transported around 70%. So here you can see the whole reaction. These are the tissue cells, the carbon dioxide dissolved in plasma 10%, combined with hemoglobin to form carbamino hemoglobin 20%, while this reaction carbon dioxide plus water gives carbonic acid. Carbonic acid dissociates into hydrogen ion and bicarbonate. So this will actually combine with hemoglobin, while bicarbonate will move out, and to take its place chloride will move in. This is called chloride shift. The moving of bicarbonate outside and movement of chloride inside. So now that was from the tissues to the blood, but from the blood to the lungs, what will happen? Now in this form 1, 2 and 3, now all these will go to the alveoli. This is occurring in pulmonary capillary. Now once they reach to the alveoli or lungs, what will happen? The first carbon dioxide in dissolved state will simply diffuse into alveoli. Why? Because the quantity of carbon dioxide is less in alveolar air as compared to blood. Referring back to our lecture or discussion when we discussed the composition, air composition in different segments, alveolar air, blood and environmental air, all airs we have discussed, the composition of all the airs. So simply diffusion will occur from the region of higher concentration to the region of lower concentration. So this is the region of lower concentration, so simply carbon dioxide will diffuse out. And second one is this carbamino hemoglobin plus carbon dioxide is that, and this is a loosely held bond, so simply carbon dioxide will move out. Number third, both of these are RBCs. Again, now chloride shift was that chloride ion moved in, bicarbonate moved out. So simply what will happen? Bicarbonate will move in, chloride will move out. So again this will make carbonic acid. Carbonic acid will again disintegrate into carbon dioxide and water, and ultimately carbon dioxide will move out. So from pulmonary artery to the pulmonary vein. So carbon dioxide dissociation curve. This depicts the dependence of total blood carbon dioxide in all its forms on PCO2. We discussed oxygen dissociation curve, so now we are going to discuss carbon dioxide dissociation curve. So this actually dissociation curve means once carbon dioxide is in the blood, how it is released into the environment. So normal PCO2 or the partial pressure of carbon dioxide ranges between 40 to 45. As we have discussed, arterial and venous. Venous blood has 45 millimeter of mercury partial pressure of carbon dioxide, while arterial blood has 40 millimeter of mercury. We have already discussed it. This is a very narrow range. It means that to play carbon dioxide has to play within this range. So that is a normal range. So gas pressure of carbon dioxide, this is the curve. So there is an effect called Helium Effect. What is that? Earlier in this chapter we pointed out that an increase in carbon dioxide in the blood causes oxygen to be displaced from the hemoglobin, the Bohr effect, which is an important factor in increasing oxygen transport. So why was that? Simply when we discussed oxygen transport, simply what happens, increase if this is the blood. So for example, this is the blood here. So cells, tissue cells, and these tissue cells are respiring. Simply, these tissue cells are respiring and these are producing carbon dioxide. What is Bohr effect? Bohr effect is that carbon dioxide from these tissue cells will move out in the blood, and as the quantity of carbon dioxide will increase, it will cause delivery of oxygen. So it will cause dissociation of oxygen from the RBC. That was the RBC. RBC had the oxygen. So this delivery of oxygen will, if this increases, so this will trigger the oxygen to be unloaded from the RBC into the tissues, because greater quantity of carbon dioxide means that cells are respiring and they need new oxygen, and new oxygen is present in RBCs. So increase in carbon dioxide will cause unloading of oxygen from the RBCs and ultimately make that RBC empty. If you have to load on a carriage, if that carriage is empty, you can reload it, simply that. So now this RBC is empty, so this will go for oxygenation and this can load, or this can carry more oxygen. So ultimately increase in carbon dioxide will cause unloading of oxygen. That was Bohr's effect. So now another effect, the reverse is also true. Binding of oxygen with hemoglobin tends to displace carbon dioxide from the blood. Indeed, this effect is called Helium Effect, simply. So now it is well understood. So respiratory exchange ratio or ventilation perfusion ratio is that normal transport of oxygen from the lungs to the tissues by each 100 ml of blood is about 5 ml, whereas normal transport of carbon dioxide from the tissues to the lungs is about 4 milliliter. So thus under normal resting conditions, only about 82% as much as carbon dioxide is expired from the lungs as oxygen is taken up by the lungs. The ratio of carbon dioxide output to oxygen uptake is called respiratory exchange ratio. Then we have two terminologies called alkalosis and acidosis. Acidemia, emia stands for blood. Acidemia means acidic blood, or acidosis is defined as increased acidity of blood pH below 7.35. So alkalemia or alkalosis is increased alkalinity of blood pH above 7.45. So there could be multiple causes like respiratory acidosis or respiratory alkalosis. So respiratory alkalosis is caused by hyperventilation, whereas respiratory acidosis is caused by hypoventilation. So due to airway obstruction or central nervous system depression or chronic lung disease. So in contrast, there are other reasons could be there. The primary cause of acid, if respiration is involved, for example, lung way stenosis, or there is a problem in lungs, as in choking, so obviously carbon dioxide will not be able to come out, so it will cause cyanosis. So increased quantity of carbon dioxide in blood is called hypercapnia. So in contrast, when the primary cause of the acid-base imbalance is metabolic, for example, due to metabolic disease or abnormal renal function, it is described as metabolic acidosis, metabolic alkalosis. So how renal function is associated with alkalosis or acidosis? Because hydrogen ion handling. So nephron have different sites to handle, specifically late nephron segments are specialized in handling hydrogen ion. So ultimately, if there is a metabolic or renal function, this can cause variability in pH, accumulation of hydrogen ions. So ultimately it can cause metabolic acidosis. So thank you very much for your attention. So for next lecture we will take up regulation of breathing. Thank you.